All right, thanks everyone for coming back after a month of, or a little longer than a month of hiatus from my travels being away. Hope everyone's been having a good summer also and taking some time off. Just some covert news. This one paper preprint here that many of you have been involved with. It was finally accepted last week, so that's and it'll be out August 8th, I think, officially. So on Science Translational Medicine. So you'll see that coming out in a couple of weeks. So that's exciting to hear. And there's other papers I think are currently under review too. There's no more updates other than, we have to get back in gear. I know Al is summer break and stuff, but the case reports that we talked about for long COVID, we're still working on that. There's a little have to catch up with that also. All right, so let's dive into the news. It's been a month since I've updated everyone. I'm sure people have been keeping track of everything. The most recent news is that probably heard in the past week or 2 weeks, couple of weeks, there's starting to be a little uptick of COVID infections. They've been maybe seeing 10% increase at the end of July 15th, which was 13 days ago, and it's still increasing, so that's a concern. There's been some increase in hospitalization in the emergency room, but not that drastic, and I'll show you those numbers later on. In this line, also, the CDC has put out, I guess, a preparation or warning saying, we should be ready because it was last year we had this happen, and the previous year, the 3-bug COVID, flu, and RSV pandemic that could come out and cause issues. I'm talking about why this uptake might be is because of this new variant emerging, EG.15, which is a subvariant of the XBB variants, Omicron. But I'll go into that in a few slides down. And the wastewater, there's been some showing the indication of that. But in that context, also, there's a nice paper that came out a couple of days ago talking about how they basically, in this paper here, they looked at wastewater metrics, and early on we knew wastewater metrics were very spot on with the actual case reporting and hospitalizations, but now the sync is not there anymore, and the whole point of this paper is showing that wastewater is probably the best metric out there because, as they put here, this text from the New York Times article in the paper itself, talking about, obviously we know now, due to the home testing, the lack of reporting, hospitalizations are not as frequent due to this variance and the natural immunity and the higher vaccination rates. Those are not very good indicators. This is the basic point of the paper. Those are not very good indicators of COVID infections in people, and wastewater is actually probably the only true left metric that people should really look at about how COVID infections are going to be increasing or decreasing. Again, another good, paper, another good endorsement for wastewater monitoring should be a common resource that should be there and a tool that people should use. This is the plot from here. We're looking at the wastewater percentages compared to the AOC curves, looking at the percentage versus either case reporting or hospitalizations, and you can see that the wastewater, the percentages over time, the sensitivity of it. In the detection, I gotta thank Steve for bringing my awareness to this one. He sent the email this morning, and I thought that this was pretty interesting. I read about it, so thanks, Steve, for sending me the update. So this is a neat. I thought this was pretty neat. This is a new device. This paper came out earlier this month when I was away. 18 days ago, but it's this air monitoring device that could detect COVID viruses in like 5 minutes. So this is from the paper. This is the schematic of what this device looks like. Basically using, HEPA filters to clean out different size particles, and it's based on detecting the size of the particle coming in, filtering it out for the size of specifically for SARS-CoV-2 virons, and then they use this nanobody derived in llamas to covalently attach to the electrode surface, and that's what they use to detect the amount of SARS-CoV-2 in the air, which is pretty clever. I came up with
quick question.
Yeah. Go
Is it
ahead.
sensitive to other kinds of viruses?
They didn't test. I'm sure they can modify that, but they didn't talk about that in the paper. So
Okay.
I'm sure they're probably looking into, because it would be useful, yes, to not only do SARS-CoV-2, but look at other ways of
Yeah. And
monitoring.
see
But
what's ambient in the background, that environment too.
yeah, yeah,
Well,
exactly.
it looks like it's actually using an antibody, so it should be pretty specific.
Yeah, for this
It's an
test is
antibody.
exactly. Yeah,
Yeah.
exactly, Dan. Yeah, exactly. So if they
So
were
it's
going to
yeah.
look up, yeah, so if they're going to look at other viruses, find an antibody for that one, like influenza or something like that, right? So yeah, so it's very specific. So these are the plots for this is like the
Gotcha.
first figure. The wet cyclone is what they're referring to here is for this method. So this is their detection method from low. They basically tested, released virons in a closed environment, contained environment, put their machine in there so they can actually know the copies that are actually being circulating in there, and then they can control it as an experiment. And they released it and they compared it to other devices and other ways of looking at like spot samplers or these biosamplers that are not aerosol based, or I should say air monitored based. And they see that there's even the low viron particles in the air. These other methods that previously people used, this is obviously you're not detecting it here and here. You see how well you could detect it. Um, Something they did also here, this plot here on the lower left, they again the closed environment with their device in there, they were testing the different variants. So they released the different variants in the air from the original Washington strain all the way to Omicron, and they were testing to see how well it was able to detect the RNA copies in the air. And you can see the detection rate. Omicron actually does better than the previous ones, and Washington was the best. So seems like a neat device and good way to monitor what's in the air. Maybe planes should use this, or restaurants or schools. I think this could be very useful. But yeah, thanks, Steve, for sending that over, because this is pretty neat to look at. So I talked about the new increase happening, this 10% increase that we've been seeing in the EG.5. There's some good Twitter posts from good scientists like this guy here, who was evolving this convergent evolution complicated chart. But you can see from the Omicron variants, obviously there's a lot of different Omicron variants that have come out. Some, of course, were outcompeted over others, as we know. And this is where EG.5 stands from the evolution of XBBs that are coming. And it's closest to this XBB 1.9.2, which we knew was coming up. Started in, was it Singapore, I think, before, and then it's spreading. So the lineages, you were curious of where it stems from and related to Omicron, is down here, and closest to these different variants that are popping up from the XBBs. So the mutations are not too far. Well, they're far enough away from the XBB where is it? I don't know. The 192 was there, but the XBB dot 14 and 5. Anyway, yeah, you just take a look at this. I'll share the slides with people. And this other scientist, Mike, who does a good job of tracking and posting on his Twitter page, he was showing that the EG.5, the Asian countries are getting that's from sequencing. They're seeing the frequency being dominant in these Asian countries, and for example, in Europe, the most dominant countries in Europe that have EG.5 is Portugal, and then Sweden and Austria are following next. So if this is the cause of the increase, those countries will probably see it first, a spike happening. But it seems like the hospitalizations are not being drastically increased, and the symptoms are not, unless someone knows more insight than I do about EG.5. But it's fairly new, so people are starting to look at it. So that's the acute COVID news, I should say, or COVID news. But the long COVID news, there's some interesting papers that have come out. Oh, go ahead.
Just a parenthetical question from Steve when you
Yeah.
were going through running back relative to the uptick a little bit. I was on with a science conversation with a colleague from Boston,
Uh-huh.
and she noted that she knows people with COVID in the past several days and everything. So I don't know what the local Boston showed, but parenthetically, since you're
Yeah,
there,
yeah.
I can.
So I'll go quickly to that. Look at this is Middlesex County where Boston is. This is the wastewater. So there's this. This is before I left. It was down here in the beginning of July. This is what happened in the 4 weeks that I've been gone, or now. So there's a huge uptake. So Steve, to your comment, yeah, it's not surprising if you're seeing a lot of people getting infected.
That's amazing that that her anecdotal comment is translated here into a more global data.
And remember, the wastewater is always 2 weeks behind what is actually going to happen because they're going to show
Hmm.
the infections first, and then people are going to report it.
That is really interesting. Thank you.
Yeah. So yes, and then you see that with BioBot, they're actually reporting the different sequences, and EG.5 is this yellow that's starting to creep up in the wastewater here.
And did you
And this
mention, I may have missed it, what the strain is, if anybody knows that's accounting for this yet, or is it multiple?
they think it's this EG.5, so
Okay.
and it stems from XBB.1.92, so it's evolutionarily coming from this side, and it's pretty far removed from these other ones that have been popping up and going away.
Okay. Thanks.
Yeah.
Action
Peop
or
I was going to say, sorry, Bill. So I was going to say people who are more familiar with the mutations, Nikki is usually my person, but I know she's on vacation. But people who are usually familiar with the mutations can maybe speak up what mutations are causing it, because I haven't read about that actually. So if someone knows about it, they can mention it.
yeah, action or maybe Adam is more in tune with his question. Have antibodies, COVID antibodies, been searched for in wastewater yet?
I'm sure it has. I mean, that's what they look at. Adam, I'm sure you could comment on this. He's on mute. We'll come back to it. We'll come back to it. Yeah, but I'm sure people have looked
Yeah,
at
I'm
that. Oh,
sorry,
sorry.
sorry, Ashton. I had to
Okay.
take myself off mute.
That's all right.
So no, I haven't seen a whole lot of, preliminary research even into looking at antibodies.
Mhm.
Primarily with the wastewater surveillance, we've been looking at, viruses, bacteria. Most recently, we've seen some, fungi that we've been looking for, fungal pathogens. And then, of course, with the chemical side of things, the narcotic indicators. But antibodies, I'm not necessarily sure if the urinary or fecal excretion is going to, if it's going to be at the signal that we would be able to quantify it within wastewater. But, it would be interesting because I was talking to a colleague yesterday that they were looking at, they had a positive spike in Kentucky for measles, actually.
Mm.
And what they ended up finding out was it wasn't a measles outbreak, but it was a signal that came from the live attenuated vaccine. They had a vaccine drive in the area. And the signal that they found in
Mm.
wastewater was actually due to the vaccination campaign that they had ongoing in that region. So, my colleague was speaking yesterday to the point that we really do need to dig in a little bit deeper into, especially diseases that are going to have a live attenuated vaccine as the mechanism of vaccination. We're going to have to dig into an indicator within wastewater a little bit deeper. So it is
Makes
possible,
sense.
but I haven't seen it done.
So were they able to verify in Kentucky that the indications of vaccine activity were due to antibodies generated by the vaccine?
By the live attenuated vaccine. Yes.
Yeah. Okay, good. Thank you. Got it.
So
Yeah, imagine.
this is Kevin.
Wait,
I
are you
have a
sure? Are
follow.
you sure about that? They were probably just measuring nucleic acid sequences.
Well, and yes, it was a nucleic acid sequence assay that they were doing. So it was the spike in nucleic acid for that for the disease was due though to the live attenuated vaccine. At least that's what he was saying when I was in discussion with him yesterday.
Right. Yeah. So the vaccine does attenuated vaccine still produces viral copies that then get their way into poop.
No, absolutely. That makes a lot of sense.
Yeah. Kevin, were you about to comment too?
Yeah, I was just curious. Have there been any studies conducted that did correlational analyses to possibly assess changes with human microbiome associated with maybe COVID-19 detection in wastewater? So, for example, are there representative microbes showing increases or decreases from the human microbiome?
You know, it's a growing field, but I'm not aware of any studies I've seen come out recently that have done an association to anything in the microbiome within wastewater epidemiology.
Okay.
Kevin, something to write a grant about.
Yeah.
Yeah. No. Yeah.
Oh, absolutely. Sign me up.
Yeah.
Okay.
Okay. So let's go on to long COVID news. There's some actually probably interesting papers for people here. This one came out. This is a Washington Post article, but this is the Lancet paper, eClinical Medicine, and this one's talking about a UK study that was looking at long COVID patients and doing computational analysis relating to 2 years out and how would it affect your cognitive issues or brain functions. And I forget how many people were in this study they had, but they had a good number of people because it's more looking at clinical records and then relating it to mental health. So this is mental conditions, physical conditions. So this is an example of figure 1 of the paper. We're looking at using these directed acyclic graphs to then make their comparisons that you see here. So basically what they had, this one final figure of the paper, they were looking at cognitive accuracy. So they give it, two different rounds of cognitive tests, which they describe in the paper what they are. I don't remember what they were. And then related to if they were asymptomatic at one point, COVID or COVID negative, comparing that to symptom duration and so on. And the whole point of the paper, they're showing that indeed some of the results they're showing, like this is the first, let's say this is the first round of cognitive, the test, round 1 and 2, so cognitive accuracy, and they're looking at principal component analysis, principal component 1 specifically. You can see like decline, the error bars are kind of big though, or standard deviation, let's say. But you can see the decline is still there, and even here some of the stuff has been improved, but there's some decline happening depending on which group you're looking at. Let's say if you look at the COVID-19 positive group that had the symptom duration for 4 to 12 weeks, 2 years later, their cognitive test and accuracy is still lower than the controls, I guess you would compare the two. So it just depends. Like asymptomatic, they have improved cognitive, or no changes, say improved, or because it's still within the. So you can see that depending on they stratified depending on how long your symptoms were, any COVID positivity, you can maybe have long-lasting cognitive decline or accuracy in your brain functions. But again, no biology here, is purely doing these cognitive tests and then going back to self-reporting.
Did they include an experimental group for cognitive treatment to either pharmacological or therapy?
No, I don't think they did that. No, I think it was, yeah. I could be wrong because I skimmed through the paper and looked up but I don't recall them having that in there.
Yeah, it's too bad they didn't do that. I mean, there's a number of therapies that could be used. For example, a lot of treatments to counter aging, cognitive aging, could have been employed, and then they could have just simply tested across conditions to see how effective those would be. But
yeah, this little node chart to show like what they've looked at, like demographics, age, sex, BMI, and things like that, but they didn't do any treatments. So Eric Topol actually had a good blog last week, about 9 days ago, about asymptomatic COVID to long COVID and pendings, and he talked about 3 papers that I had missed that you guys probably noticed. And this one paper here was looking at HLA is associated, basically, as the title says, asymptomatic SARS-CoV-2 infections and how this might impact long COVID. So basically, these authors, this is taken from Eric Topol's blog, since he did a nice job summarizing the papers. Or this one paper, and there'll be a couple more papers to look at. But basically, they looked at nearly, there's a large cohort, 30,000 participants, so that was a pretty large cohort of people to look at. and they were using basically ways to track the COVID symptoms was through the smartphone app to study the tracking. And then what they did, they looked at the biology associated with that. And I don't think enough 3,000, but they looked at over 1,400 asymptomatic positive tests, I guess, from that. So basically what they found was this HLA locus was strongly associated with symptoms, asymptomatic specifically. And people, I'm not very familiar with HLA alleles and their functions and everything, but I was just reading about it here. The specific one, NQK-Q8, it might mean something to people here, but specifically for SARS-CoV-2 asymptomatic folks, it was related to like the T cell functions, and the response was seeming to be active there. So basically, this is paragraphs from actually the discussion of the paper, and what they saw, they actually then also compared it to other coronaviruses, see if this is SARS-CoV-2 specific or coronavirus specific. And one thing that was, I guess, kind of neat to see is that this thing I bolded and underlined that I saw, and I probably missed some of the key factors of the paper too, because I didn't read it too in depth. But they're saying the results, support their hypothesis that this HLA alleles locus here mediates asymptomatic disease, and is found to actually be potentially pre-existing T cell immunity due to other coronavirus infections. So maybe it could explain, if you're asymptomatic, was it because you had other coronavirus infections related to this HLA previously mediated through this other. That's what I gathered from it, and reading this, and the T cell cross reactivity to this. But if other people have read this paper and I've missed details, please speak up. But it's or know more about HLA alleles and functions. I don't know if there's any insight from the group here on this. But it could also be related to like immune functions and things like that. That could be maybe even a discussion point to add in our inflammation paper we're doing. This was an interesting paper here. And there's a couple of preprints he actually talked about really long COVID. So this one preprint that just came out, I think last week or this past week, last week, by the genome-wide association study for long COVID. So they basically looked at this, did a GWAS study, looking at over 6,000 people with long COVID, and then looking at different studies over 16 countries. The one caveat for this data, they're looking at mainly European countries. So the one limitation is the diversity is European population based as opposed to the rest of the world. So I'll keep that in mind if that changes it. But what they found here doing this GWAS study is this one gene, FOXP4 transcription factor, seemed to be expressed broadly in the body of a lot of tissues related to long COVID. So that was interesting to see. And it's chromosome region 6, if that means anything to people, or might be insightful for people to see what that happens. But this is the basic point of this paper. There's a lot more data and a lot more analysis they did, but I'm not familiar with this gene. So if people know what this gene does, please let me know or speak up here. But this is interesting to see. So maybe something we go back and look at with our data we have, and how this potential gene could be a link to long COVID and what it does. Again, I didn't read the paper in depth, but in the discussion,
I just looked it up. Looked up FOXP4 in my single cell atlas. And it's in definitely functionally associated with goblet cells in tracheobronchial, which is one of the cell types that we thought that even I thought were different in African Americans versus Caucasians. The proportion of goblet cells, but it's also pretty highly expressed in dendritic cells in the lung. Yeah, CCR7 positive dendritic cells.
That's interesting.
Is probably also non-classical monocytes. So those are two really hot effector T cell or myeloid cell types that really modify response to virus. And
so like
things going on here. One is the asymptomatic nature of the initial COVID infection, and the second is the risk of that going to long COVID.
seems to have a heavy immune component for this gene.
Well,
or
I mean,
involvement.
those two are non-classical monocytes, and
Uh-huh.
dendritic cells are two gigantic effectors of immune function.
Right.
And do you learn? You go from innate immunity to acquired immunity. That's very dependent on dendritic cell function and HLA presentation.
Oh, that's okay.
So FOXP4 would be a potential strong modifier of dendritic cell function.
Mhm.
How well you teach T cells that. But what did they say in the paper? Did they get any of the other?
No, I mean, again, I didn't read it in depth, but they did basically they're focused on what this gene because it was like the major one that popped up from their GWAS analysis. There was other genes in there, obviously, and other locuses that they could look at, but this is the one that was like the screaming, like, I guess the red alert for long COVID and what popped up from their analysis from the European community. They have the data. I think they have a link for their data somewhere, so we can look at other genes that might be related and involved with their study.
I'm wondering if they did. Did it not hold up if in the presence of symptomatic COVID risk of getting long COVID?
Oh yeah, the stratification between asymptomatic and symptomatic. I don't. Again, I have to go back and read the paper, so I'm not sure if they did that. But they were looking at mainly the long COVID population. I'm sure they summarized somewhere in here their symptoms they had. I have to go back to the paper and look. I'll share the paper with you guys. Yeah, maybe I'll do that now in the chat, just in case you want to see that information.
Okay, cool.
So here's the paper in the chat. So take a look, because as I said, I didn't have time to read it in depth, but they have all the patient population data and everything else in there. And it's a preprint too. Remember, take it for a grain of salt. So have to review these things, but it seemed pretty solid. And another preprint here, they're looking at genetic factors related to fatigue and what's dominant for long COVID, and then related, the CSF and ME symptoms that we know related with chronic fatigue syndrome and so on. So this preprint here that came out again about 15 days ago, a couple of weeks ago. This cohort of patients wasn't as much. I think they had in the hundreds. I forget. I have to go back and look. But what they did was they used some computational tools and they stratified using these, kind of this network analysis and structures diagrams to compare severe or fatigue dominant long COVID patient populations from this platform that they were using. And then
This Bill Afshin. Is it fair to say in this chart here, this cross tab, that the frequency of mental or behavioral disease and declining down the list there is correlated, going up with mitochondrial density in the involved tissues?
I haven't got to that point yet, but not necessarily, because that's not what they looked at in the study. So what they basically looked at was the SNP genotypes and disease associated with that, and then related it back to population pathways and other factors related. Yes, there are metabolic for severe. This is severe here, for example, this dot plot, and fatigue, long COVID separation for the patient population. And you could find things related to mitochondria, this gene ontology pathway analysis they did, things related, immune related functions, metabolic processes, and then this heat map here again is showing different stratification and different pathways that might be related to the different long COVID genes in this cohort that they identified. And yes, there are probably things related to mitochondrial activity, but no, that's not what they're looking at.
Okay, thanks.
So this is an interesting preprint to look at. Again, this data set here is a lot more comprehensive than this one, but again, Eric was talking about how it could be related to asymptomatic cases versus long COVID and so on. So those are the papers I've seen. I probably missed a bunch of other papers. If there's more papers people can mention. One thing I think Diego had put in the comment, let me show you this, I discussed this when I saw him too, is that for future pandemics and things like, and the future risk of virus spreading, this was the thing that WHO put out, what was this, 2 weeks ago? But this is actually from April 5th, an article, but they talked about it again a few weeks ago, talking about the risk of dengue, Zika, and other things outbreaking during the summer heat wave. This is in April, but Diego had brought up in a meeting earlier, and you shared some of the articles, Diego, nicely, where they've been reporting, actually, I think Nydia was there too, Nydia was talking about this too. Dengue has been seen around Europe now based on the global warming that we see and the heat changes happening. And even Diego pointed out there's been a couple of cases of malaria popping up in Texas. So that's something for pandemic preparedness and using our tools to keep an eye out, because due to global warming and the extreme fluctuations of heat, the mosquitoes that are carrying this disease are actually getting to places that were previously not been able to get to. But I thought that's how you chat, Diego, so I thought I'd talk about that really briefly. I don't know, Diego, if you want to comment a little bit on that. Additional insight?
No, I just thought that was important to consider since these diseases are really nasty. So I am based in South America and I have experienced them firsthand. So yeah, that's why I raised this concern also to the guys in covered and also in the other projects that we are working on. Yeah.
And you mentioned, and then I didn't realize it until you mentioned WHO actually making some legitimacy, because WHO is saying people should be better to pay attention to this, because this is something that could be drastic if it does continue this way.
Exactly.
I've often wondered what the full spectrum of mosquito activity is, because that mosquito activity is very much larger than the presentation of morbid conditions in any set of individuals anywhere. And you got to wonder whether mosquitoes actually convey antibodies from one person to another as they go from one person to another. You know,
well, let's not speculate on that, because we have no idea. So I have no idea on that. We could
others can work on that
offline. Yeah, we can talk later offline on this. So let's go to the tracking of variants. So as I said, EG.5 is that variant that's coming up now, and before it was, XBB.16.15 are those variants that are, this is the US now, but around the world, the same thing. But you can see EG.5.1 is the one that's being attributed to this increase, and you can see places like California, and we see an uptake in California too. These are being dominant. And I showed this earlier when Steve asked the question. The wastewater overall, you can see this is nationally in the US, you can see it's going up. In some regions, if you actually go in, and this is what Steve brought up in Boston, this is Middlesex County's Boston, you can see this big uptake, which is a lot more than 10%. So depending on the region you are, if you look at this BioBot and their wastewater, look at your county, you might be lower, you might be like this. So maybe wearing masks inside should be a good idea. Definitely on planes, probably. And unfortunately, again, I guess I don't have a good monitoring website to look at globally that has this nice perspective. And I think, Adam, you said there's few out there, but not as nice as BioBot. So people know good sites that show like South America, Europe, and Asia wastewater like BioBot does. Please send it my way, so then I can keep track that way and share it with you guys.
I'll look into it, Afshin. I do know that, certainly in South America, Africa, the number of even studies that are doing this type of analysis are not zero, but, nothing compared to the response that we've seen here in the US or Europe.
Right.
So we're working with a group in Malawi right now to develop a cost-effective wastewater surveillance platform, with the hope
Nice.
that over the next year or two that we can get something that's going to be effective and cost-effective for developing nations to cover that 83% of the population that resides in developing nations. At that point, then we can, I think, approach wastewater surveillance from a global surveillance perspective.
Nice. Yeah. Well, look forward to that if you get it going. It's probably a long effort and a lot of work, but worth it probably once it's done.
I would agree.
Yeah. So the variants in the U.S. again, going back to another way of looking at it, the CDC tracker, you could see how we've seen this evolution happen many times over the past 3.5 years. Now XBB.1.5 was this, but slowly you can see these other variants, the different colors that were coming up and down. Some were XBB associated with the 1.16, one were for the 1.92 as we saw. But now EG.5 seems to be spreading rather quickly. And depending on the region you are here, it's this mustard yellow color. You can see California has more. Here's the Northeast here. You can see that, like where Massachusetts or New York and Pennsylvania, you can see, and Maryland, you can see that it's starting to creep up pretty quickly. So right now it says it's between 8 to 15% of the cases in the U.S. from their sequencing they do. But I imagine if it does anything like we've seen previously with these ones that do dominate and take over, this one will be the next one, EG.5, that people will see instead of, what is it, blue, navy blue here, that'd be mustard color coming up next. And then globally, again, as we talked about in that paper that I showed you initially, this site here, ourworld.indata.org, was really nice in the beginning of the pandemic when there's a lot of monitoring happening, but unfortunately, as we know, and what the papers have shown now, this is not going to be as accurate to look at and monitor. And even like hospital admissions is not a very good indication of COVID infections because, as we know, through the immunities of people getting sick often and developing immunities and the vaccination, and due to the virus itself is not causing hospitalizations as much as of course the first versions of it. So this is not a very good indication of what's happening in infections, but we do see some uptakes happening even despite that. South Korea, for example, might be doing a better surveillance program still than other countries. I'm not sure, but you can see in South Korea it's starting to go up. And we saw from the earlier post at EG.5 in Asian countries. Let me go back up here. Oops, here it is. South Korea is this purple color. It's right here. The percentages or frequency is like 20% now for South Korea. So it's pretty high up compared to growing, I should say, in the Asian countries. So this is not surprising. But the hospital's admissions for the countries who do keep track, South Korea seems to have stopped reporting now at the beginning of the year. But United States still reports, I guess, hospital admissions for their COVID. So you can see that's pretty flat. Or patient ICUs. You can see Japan and US are still reporting here. So that's the current news and updates. Thanks for people coming on after a long hiatus.